Once we have discussed the cell-derived mediators, we are now going to move on to the second important category, that is of the plasma-derived mediators. And please recall, I told you, although they are circulating in the plasma, they are being released from which organ? That is the liver. And they have these three similar sounding pathways, that is the kinin, clotting and complement. So in the same order, we are going to begin first with the kinin system. Now what do we mean by this kinin system? It will result in the formation of something of the kinin family. Let's see, whenever there is any injury, there is activation of factor number 12. Students, please recall, factor 12 is also known as the Hagman factor. So on injury, this factor 12 gets converted into its active form, that is 12A, A for active form. This factor 12A is then going to convert prekallikrein into kallikrein. That in turn is going to convert kininogen into bradykinin. So students, we have ultimately reached the synthesis of a kinin. Whenever there is any injurious agent, if there is any injury that happens, the first thing that gets activated is the Hagman factor, factor 12. On activation, it converts prekallikrein to kallikrein, which in turn is going to convert kininogen into bradykinin. So this is the kinin pathway. What you need to know, which factor, which coagulation or clotting factor is responsible, that is factor number 12, Hagman factor. Next, you need to know what are the functions of bradykinin. What are the important functions of bradykinin? So remember that bradykinin is the one that is responsible for venular dilation. So vasodilation is one function. Particularly venular dilation is very common action of, or in fact the most important action of bradykinin. Other than this, it is also involved in the pain. We have done other mediators earlier for pain, which were the prostaglandins, D2, E2, F2 alpha, particularly E2. Next we are doing for pain is bradykinin, and it also causes bronchoconstriction. But out of these three, if someone asks us the most important action of bradykinin, then the most important action will be venular dilation. Now again, let's try to link this up with a little bit of pharmacology. What happens in the body is that bradykinin is going to be broken down into its metabolites by an enzyme called ACE, angiotensin converting enzyme, right? What is angiotensin converting enzyme normally doing in our body? It converts angiotensin 1 into angiotensin 2. What is angiotensin 2 doing? This activates the renin, angiotensin, the aldosterone secretion is activated. Sodium water retention results in elevation of blood pressure, hypertension. So what type of a drug can you give to a patient of hypertension? ACE inhibitor. We are going to inhibit this and thereby result in decrease in BP, because an angiotensin 1 will not get converted to angiotensin 2, aldosterone will not be secreted or will not be resulting in sodium water retention, so the blood pressure will go back to normal. But in the process, when we give ACE inhibitors, we are definitely curing the patient of blood pressure, we are getting the blood pressure back to normal. But in that process, will bradykinin now convert to its metabolites? No. This process will not occur, resulting in elevation of bradykinin. Now please recall, bradykinin when elevated what all actions can it have? We know that bradykinin can first of all cause bronchoconstriction. So the most common side effect will be dry cough. The most common side effect will be dry cough because bradykinin causes bronchoconstriction. Please recall, we just did the function. Bradykinin causes bronchoconstriction. So when more of bradykinin is there in the body because the patient is on ACE inhibitors, bradykinin cannot be broken down into its metabolites. This bradykinin is going to result in dry cough. Next, because the most important action of bradykinin was venular dilation, this will also result in angioedema. So students, when bradykinin levels increase, patient can suffer from dry cough, angioedema. Indirectly, I can say the side effects of ACE inhibitors will be dry cough and angioedema, and that is happening because of bradykinin. So this, students, was about the kinin system. Particularly in the kinin system, we are referring to bradykinin. The most important action of bradykinin is venular dilation, followed by pain and bronchoconstriction. The next, students, that we are starting with is the clotting system. Again, a very important mediator that we have is the clotting system. Now what we need to know over here is that whenever the clotting system is activated, there are two pathways that it can occur via. The coagulation or the clotting system comprises of two pathways. One is known as the intrinsic pathway. One is the intrinsic pathway and the other is the extrinsic pathway. The other is the extrinsic pathway. What we need to know over here, students, is that both the pathways will have their own set of clotting factors, but ultimately they will come and converge onto one point from which everything else will be common, and then we will call it the common pathway. So first let's see their individual paths and then we'll merge them and get to the common pathway. So what is the intrinsic factor? How are we going to remember? So the entire pathway, whether you're dealing with intrinsic, extrinsic, remember the mnemonic 10. So let's write down 10 over here, T-E-N. This means the first factor that will get activated in the intrinsic pathway will be the factor number 12. When it gets activated, I will call it 12A. This in turn will go and then activate factor number 11. So T was for 12, then E is for 11. 11 into 11A. That in turn will go and then activate factor 9 into 9A. So 10, 12, 11, 9. That is how we are going to remember. This 9 that has been activated into 9A will then go and act and convert factor 10 into 10A. And in doing so, it also uses factor number 8 and calcium. Factor number 8 and calcium. Please note, calcium is nothing but factor 4. Calcium is nothing but the other name for factor number 4. So we have done the intrinsic pathway. 10 is what you need to remember. T-E-N, and finally you have to come on to 10, factor 10. So T-E-N means 12, 11, 9. Finally come to the number 10. That was about the intrinsic pathway. What about the extrinsic pathway? Now in the extrinsic pathway, again we are going to take the same mnemonic of 10. But over here we are going to add a little bit of mathematics to it, and this will include two factors, that is 7 plus 3. So 7 plus 3 also amounts to 10. Factor number 7 and factor number 3 are the two factors that are going to play a role in the extrinsic pathway, resulting in the activation of factor 10 into 10A. So ultimately we have dealt with two pathways, the intrinsic and the extrinsic pathway. Both of them are using the same mnemonic, that is 10. For the intrinsic pathway, TEN, that is 12, 11, 9, coming on to a common factor, that is 10. For the extrinsic pathway also we have the mnemonic 10, that is factor 7 plus 3, that is again a 10, which will converge onto activating factor 10. Now that both the intrinsic and the extrinsic pathways have converged onto a common factor, that is factor 10, and activated it into 10A, what we have now onwards will be classified as the common pathway. It will be classified as the common pathway. Now students, this 10A that has now entered the common pathway, this 10A will now convert prothrombin to thrombin, which in turn will convert fibrinogen into fibrin. Now there are few more factors that we need to consider over here. When factor 10A was converting prothrombin into thrombin, what coagulation factor is it? Factor number 2. When 2 gets converted into 2A, again, till now the mnemonic was that in intrinsic and extrinsic we were dealing with something to do with the mnemonic 10 that was coming to a common factor 10. After coming to 10, let's start breaking this down for our memorizing purposes. So if we divide 10 by 5, we will then come to a 2. So factor 10A in the presence of factor 5 and calcium converts prothrombin into thrombin. This thrombin, which is factor 2A, then converts clotting factor 1, that is fibrinogen, into 1A. What is the main aim of doing this is because fibrinogen is going to be a soluble form. We know fibrinogen is circulating in the blood, it is a soluble form. This will be converted into an insoluble form. Why? Because our main motive is that in the clotting pathway, we are going to result in the form. We are making sure that a clot is formed. Clot needs to be something that is insoluble. But fibrin alone is not sufficient. This clot is then acted upon by factor 13, which is the fibrin stabilizing factor. Fibrin stabilizing factor, which is finally resulting in the formation of a clot. Now the clot that is formed will be very firm, will be insoluble because a stabilizing factor has acted upon it. So we have two particular pathways, intrinsic and extrinsic. The mnemonic for throughout this pathway has been 10. Now what we need to know is one pathway that we haven't dealt with, or you can say one clotting factor that we haven't dealt with, is clotting factor 6. So remember students, there is no clotting factor 6. There is nothing known as clotting factor 6. Clotting factors are going to range from 1 up till 13, but there is going to be no clotting factor 6. This does not exist. So if the option has any of these, then please be vigilant. There is no clotting factor 6. Now what is the main aim of knowing about all of this? First of all, what is the clinical significance? So the clinical significance of knowing about this is that certain deficiencies of these clotting factors, if any clotting factor, say clotting factor 8, clotting factor 9, if any clotting factor is deficient, clot formation will not occur, patient will have bleeding manifestations. So students, for example, if there is a deficiency of clotting factor number 8, this is going to result in a disease known as hemophilia A. If there is a deficiency of the next clotting factor, that is clotting factor number 9, this will result in hemophilia B. Similarly, there is also something known as hemophilia C, and convert the C and add one more line here. So I'm converting it into an E. So that is deficiency of factor number 11. That is deficiency of factor number 11. Next, there is also something known as parahemophilia. Parahemophilia. And parahemophilia is nothing but the deficiency of, it is the deficiency of factor 5. How will we remember it? P for para and 5 happens to be panch. So P for that and P for para. So deficiency of factor 5 becomes parahemophilia. Convert the C into an E. So deficiency of 11 becomes hemophilia C. What we already know, hemophilia A and B are deficiencies of 8 and 9 respectively. There is another clinical significance that we need to know over here is the role of calcium. Now students, calcium is what factor number? It is factor number 4. Remember, all the clotting factors are being produced by the liver except calcium. Calcium is not produced by the liver, we know. So all the clotting factors are going to come from the liver except calcium. Calcium will not come from the liver. It is not synthesized or produced in the liver. One more thing about calcium that we need to know. If we take out calcium from the clotting system, if we take out calcium from this pathway, will clot formation occur? No. So that is a very important role of calcium that is utilized in anticoagulants. Anticoagulants are basically calcium chelating agents. What do they do? When we take a sample of blood and we add an anticoagulant to it, the blood will not clot. In which scenario do you want the blood should not clot? For example, in blood banking. You do not want the blood to clot, right? So what are you going to do? We are going to add certain anticoagulants like acid citrate dextrose, that is one example. Another example of an anticoagulant is CPDA, that is citrate phosphate dextrose adenine. And another one that we have is SAGM, that is saline adenine glucose mannitol. So these three are examples of anticoagulants, and although SAGM does not have it in its name, but it also has citrate and phosphate. It is also going to have citrate and phosphate. What is common to all of these is all of them are having citrate in them. Why? Because this citrate is then going to pull out all the calcium that is present in the blood. So they are going to result in calcium chelation. Now once the calcium is going to be taken out from the blood, will the blood coagulate? No. And that is how the blood bank is functioning. That is how bags are stored in the blood bank. Whatever is collected from the donor is mixed with either of these anticoagulants, and that blood will not clot till the shelf life of the blood is valid. So this is one very important role of calcium that you need to know. After the role of calcium, there is another clinical significance that we need to know regarding the role of vitamin K. Now vitamin K is one vitamin that is very essential for certain transformations, or you can say modifications, that have to happen in the clotting factors. They are required for the gamma carboxylation, of glutamate. What is this? For the clotting factors 2, 7, 9, 10. You have to remember these numbers. Clotting factors 2, 7, 9, 10 will be vitamin K dependent. How are they dependent on vitamin K? Because vitamin K is needed for the gamma carboxylation of the glutamate residues in these clotting factors. So, vitamin K is needed for the gamma carboxylation of glutamate in factors 2, 7, 9 and 10. That is the role of vitamin K. Before we end this pathway, one thing that we need to study is laboratory tests. Although this will be covered in detail when we are dealing with platelets, but a brief overview of the laboratory tests that you need to know. Just the names for the time being. So remember, we have two tests, that is PT and the other is APTT. PT stands for prothrombin time and APTT stands for activated partial thromboplastin time. Now remember which test is for which pathway. So we will remember it by the mnemonic PET. PT, prothrombin time, is for the extrinsic pathway. Prothrombin time is for the extrinsic pathway. So automatically APTT will be a measure of the intrinsic pathway. Again, this is a question that has commonly been asked in the previous years as well. And this is also one of those questions where the students tend to confuse that which test is for which pathway. So now you are going to remember a mnemonic that is known as PET, where P and T are for prothrombin time and the E refers to extrinsic pathway. We will be discussing these tests in greater detail with how the sample is collected and how the test is performed when we deal with platelet. We have discussed kinin system. We have also discussed the clotting system. The last system that we are left with now is the complement system. So this is what we need to know next. We need to study the complement system. Now, first of all, what I want to tell you, or what you already know, I want to revise, is that complement system is something that is circulating in the plasma. It is a plasma derived mediator. There are total 20 complement proteins. Total 20 complement proteins that are present. And of interest for us in this discussion is C1 to C9. C1 to C9 is important for us. Now let's see what are we dealing with in the complement system. Remember, the complement system has three pathways. The complement system has three pathways. First pathway is known as the classical pathway. Classical pathway. Second is known as the alternate pathway. And third is referred to as the mannose binding lectin pathway. Three pathways that we have, all of them are going to have their own set of factors which will activate them. Classical pathway, first and foremost, the first difference that you need to know, classical pathway will be activated by immune complexes. What is an immune complex? Immune complex is nothing but antigen-antibody interaction. Particularly, remember, antibodies IgM followed by IgG. So classical pathway will be activated primarily by the antibodies IgM and IgG. Alternate pathway is going to get activated. So we have A for alternate, and this is going to get activated by which immunoglobulin? IgA. So A for alternate, A for IgA, immunoglobulin A. Other than this, it is also activated by the microbial endotoxins. Lastly, we have the mannose binding lectin pathway. Where have we otherwise read mannose? Mannose is present in the, I hope you can recall, the bacterial cell wall. So the mannose binding lectin pathway is going to get activated in response to microbes which contain mannose. So we have these three pathways: classical, alternate and mannose binding lectin. You know how each one of them is activated. The question that has been asked, classical pathway is activated maximally because of which immunoglobulin? Remember, IgM is more common. Alternate pathway is activated by which immunoglobulin? Remember, IgA, A for IgA, A for alternate. Mannose binding lectin is self-explanatory, mannose in the microbes. Now let's come to the actual pathway. What is happening in the classical pathway? So in the classical pathway, once it gets activated, the first complement protein that will get activated is C1. C1 will convert into activated C1. So the classical pathway is going to start in a classical way from the first complement protein C1 and is then going to get converted into activated C1. Following this activated C1 is then going to activate C4 and C2 to form a complex C4b2A. Following this C4b2A is then going to go and cause breakdown of C3. Breakdown of C3. We are going to stop here and we are going to first look at the other pathway, which is the alternate pathway. Alternate pathway we know was activated by IgA and endotoxin. What is it going to activate? So the classical pathway started from C1. The alternate pathway is not classical. It is going to take an alternate route and will start from C3. C3 in the presence of something known as factor B and properdin is going to result in the formation of C3b BB. So C3 can be C3A, C3B, it is forming C3b, and factor B can form BB. This also is going to then later on cause C3 breakdown. So the two pathways have converged. Classical starting from C1, activated to C1 activated form, will then activate C4 and C2, which will then break down C3. Alternate starts from C3, uses factor B, ultimately activates C3. So the converging point of both will be C3 breakdown that it has to do. The third pathway that we had was the mannose binding lectin pathway. Now students, there is nothing very different in this pathway. This is also like the classical pathway starting from C1, is going to get converted into activated C1 and is then going to take the same route as the classical pathway that we had, that it is going to then go and activate C4 and C2. So everything is the same as the classical pathway, only the trigger point is different. In classical pathway, it was immune complexes like IgM, IgG. Over here, it is the mannose. Ultimately, all of them have finally converged onto causing C3 breakdown. Okay, now this C3 breakdown, we know that all the complement proteins can be broken down into A and B. So the C3 breakdown will occur into C3A and C3B. C3A, A for away, A for anaphylotoxin. C3A acts as an anaphylotoxin. It goes away, it acts like an anaphylotoxin. C3B, along with all the complexes that have been formed, the next step that we are going to go towards is C5 breakdown. And this C5 breakdown will occur again into C5A and C5B. We know A again will go away, it will act as an anaphylotoxin. Anywhere else where we have read about C5A? Remember we did a mnemonic LIC for chemotactic agents. So C was for C5A, this is a chemotactic agent. It goes away, it can act as a chemotactic agent, it can act as an anaphylotoxin. C5B, on the other hand, is one very social person because this is then going to combine with C6, C7, C8, C9 to form a complex C5B to C9. All of them will join together. So C5B, 6, 7, 8, 9. All of them will join together and result in the formation of something known as MAC. That is membrane attack complex. Membrane attack complex. So students, we had three pathways. Classical starts from C1. Manos also starts from C1. Only the alternate pathway takes an alternate route and starts from C3. Ultimately, all converge on breaking down C3, which ultimately breaks down C5, and we finally have a complex of C5B to C9, which is known as membrane attack complex. Now, whose membrane is it actually attacking? So, if this is a bacteria, for example, it is the membrane of the bacteria that it is going to attack. Once that is attacked, it is going to result in lysis of the bacteria. So, the membrane of the bacteria is going to be attacked, resulting in bacterial lysis. So, this was a summary of the complement pathway that we have. If we want to take down few important MCQs from this pathway, question number 1. What are the anaphylotoxins that we have in this pathway? So remember, A for anaphylotoxin, and we have C3A, C5A. Question number 2. What are the chemotactic agent in this pathway? So I hope you remember the mnemonic LIC, where C was for C5A. Question number 3, also what we have discussed. What are the opsonins present in this pathway? Please recall, the coating of a microbe was by opsonins, process known as opsonization, that was making the organism tasty for phagocytosis. This opsonin, if you can recall, were all the B molecules like C3B, C4B, C5B. And lastly, the question that comes is, what is the composition of MAC, membrane attack complex, and that is C5B to C9. So these are the 4 important MCQs that come out of this particular pathway. Now, once we have dealt with the main portion of this pathway, now we need to know certain subheadings and certain topics that are also of interest. There are certain diseases that are associated if there is a deficiency of any of these complement proteins. So, the next part of this particular complement system would be deficiencies. So remember, if there is deficiency of C1, C2 or C4, not 3, 1, 2 and 4. If there is deficiency of 1, 2 and 4, the person can suffer from autoimmune diseases. Autoimmune diseases. So C1, C2, C4. 1, 2, 4. If there is deficiency of C3, the person will suffer from severe pyogenic infections. Severe pyogenic infections. If there is deficiency of C5b, C6, C7, C8, that is, we are talking about the membrane attack complex, then the person can suffer from infections such as Neisseria, Toxoplasma, Pneumococcus. So out of the entire membrane attack complex, I have missed out one protein, C9. I have not included that because remember that C9, if there is a deficiency of C9, there would be no disease. Even if C9 is deficient, the other proteins are potent enough to form a membrane attack complex on their own. So still then also the patient will be able to fight infections and there will not be any problem. But if the other members of the membrane attack complex are deficient, then infections with capsulated organisms like Neisseria, Pneumococcus, these are relatively higher. But overall, if you are asked which is the most common complement deficiency in humans, which is the most common complement deficiency, that is going to be the C2. C2 deficiency is the most common complement deficiency that is seen in the human population. So this was about if the complement proteins are deficient. Now we are going to do the last subheading of the complement system, which is going to be regulation. So regulation of the complement system. Now there are certain proteins which are going to regulate the complement system, and the first one that we have amongst those is going to be the C1 inhibitor. The C1 inhibitor. What does that mean? That if this is C1 and on activation it becomes activated C1. On activation it becomes activated C1, right? So there is an inhibitor which can stop this. When the body is not in need of activating the complement system, the C1 inhibitor will come and stop this step. Complement activation will not occur when it is not needed. The problem then arises if the person suffers from C1 inhibitor deficiency. If there is a C1 inhibitor deficiency, what will happen? This pathway will go on forever. There is no control left. More of activated C1, there is more of complement proteins. The complement pathway will keep on getting activated. There will be increased forever activation. There is no stopping the complement pathway because the inhibitor protein is absent. This, students, then results in a disease. When there is C1 inhibitor deficiency, it is known as hereditary angioneurotic edema. Angioneurotic edema. HAE is how it is very commonly abbreviated. Hereditary angioneurotic edema. Now students, this condition is more commonly seen in females than males. And if we are saying edema, remember this tends to occur more in the oral cavity and the larynx. Now what you can easily visualize or imagine is that if there is edema in the larynx, that definitely makes it a life-threatening condition and definitely calls for a medical emergency. What you need to know further is that edema of the oral cavity and the larynx that occurs is of the non-pitting type. Now, this was the first mechanism by which we are regulating the complement system, C1 inhibitor. If there is a C1 inhibitor deficiency, patient lands up with hereditary angioneurotic edema. The second regulatory protein that we need to know is CD46, factor H and factor I. Remember that these three molecules that I have written over here, this will regulate the alternate pathway. Till now we had done C1 inhibitor that was regulating C1, that is the classical pathway. These three molecules, CD46, factor H and factor I, will regulate the alternate pathway. Now the problem arises that if there is a deficiency of these, alternate pathway will no longer be regulated, the regulatory action is lost. So there is continuous activation of alternate pathway. There is continuous activation of the alternate pathway. As a result of this, as more and more alternate pathway keeps on getting activated, finally the blood vessels are going to suffer from endothelial cell damage. There is endothelial cell damage, as a result of which it lands up in a disease known as atypical HUS, that is hemolytic uremic syndrome. Hemolytic uremic syndrome. So to recall, CD46, factor H, factor I. These are the factors which are controlling the alternate pathway. If their control is lost because of their deficiency, if this control is lost, the alternate pathway will keep on getting activated. Endothelial cell damage will occur. This will land up in a disease known as atypical hemolytic uremic syndrome. The third and the last regulation that we need to know of involves two proteins, that is CD55 and CD59. These are the proteins that we will be dealing in great detail when we come to the chapter of red blood cells in hematology. But for the time being, you need to know that these two molecules, for example, if this is an RBC, this is where CD55 and CD59 are present. They are like anchors on the surface. They are going to have a protective role. So they are going to protect the RBC. Not only the RBCs, they will also protect the WBC and they will also protect the platelet. So CD55 and CD59 have a protective role for the RBC, WBC and a platelet. It is going to act as a shield and protect it. Problem arises that if there is a deficiency, then the complement pathway will get activated. There is no control left over the complement pathway. The complement pathway control is lost. So now the complement pathway will go and attack the RBCs, WBCs and platelets. Earlier they were shielded by CD55 and 59, but they are not present. So this control is lost, resulting in decrease in RBC, WBC as well as platelets. Particularly, there is a condition in which this deficiency is seen known as PNH, paroxysmal nocturnal hemoglobinuria. Hemoglobinuria. PNH, paroxysmal nocturnal hemoglobinuria. So this is happening because of a deficiency of CD55 and CD59. So this is another disease that we will be dealing with in great detail, but you should know that paroxysmal means intermittent, nocturnal means night time, Hemoglobin, as we already know, self-explanatory. Urea means in the urine. So there are intermittent attacks in which the patient tends to pass red colored urine at night. That is paroxysmal nocturnal hemoglobinuria. And why is that happening? Because RBC breakdown is happening by the complement pathway. Why? Because the protectors, that is CD55 and CD59, are deficient. So these are the three things, the three main points that were regulating our complement system, and I wish to recall these because these are favorite with the examiners. The first one was C1 inhibitor deficiency, resulting in hereditary angioneurotic edema, a non-pitting edema which, if involves the larynx, is life-threatening. The second one was CD46, factor H and factor I. This regulates the alternate pathway. If this regulation is lost because of a deficiency, it results in a disease called atypical hemolytic uremic syndrome. Lastly, we had regulators, that is CD55 and CD59. If these are deficient, the shield over the RBCs is lost, the complement goes and breaks them, resulting in a disease known as paroxysmal nocturnal hemoglobinuria. So students, getting back to our main list that we were dealing with, we have also finished the complement pathway. So broadly, the mediators were either derived from the cell or from the plasma. From the cell, they could either be preformed like histamine, serotonin and lysosomal enzymes. From the newly synthesized, the most important ones were arachidonic acid metabolites, which fetches maximum questions, followed by cytokines and chemokines. Plasma derived is a separate category in which the derivation has happened from the liver and it is circulating in the plasma. This has all the similar sounding terms like kinin, clotting, complement. In kinin, we had bradykinin, which was very important, resulting in venular dilation, pain and bronchoconstriction. Clotting system, which had two pathways, intrinsic and extrinsic. The mnemonic that we have read for this is 10. And remember, there are two tests, PT and APTT. PT is for the extrinsic pathway, APTT is for the intrinsic pathway. And lastly, we have the complement system. There are three pathways, classical, alternate and mannose. Ultimately results in the formation of membrane attack complex, which is C5b, 6, 7, 8 and 9. The most common out of all that can be deficient is C2 in humans. And lastly, do revise, whenever you do this topic, definitely do not forget to revise all the regulatory mechanisms that are involved in this complement pathway.